Good evening, everybody, and welcome to the University of Sydney for tonight's public lecture. I'm Alice Motion. I'm an associate professor in the School of Chemistry and deputy head of school there. But tonight I'm going to be introducing a fantastic biologist who I met on a recent trip to Canada, and I'm really excited for you to hear from Professor Maydianne Andrade this evening. Before we start tonight's public lecture and I give you an introduction to Maydianne, I'd like to take a moment to acknowledge the country that we're on this evening. We're on the lands of the Gadigal people of the Eora Nation here, and this always was and always will be Aboriginal land. There is no place in Australia, water, land, sea or air, that has not been known, nurtured or loved by Aboriginal and Torres Strait Islander people. And it's going to be fantastic to hear from a visitor about some of her experiences working in and around the Gadigal lands and beyond, I think, during her time here in Sydney. So I won't take too long to introduce Professor Maydianne Andrade, because she has a lot to share with you, but she's also an extremely accomplished academic and science communicator, so I do want to tell you just a little bit about her background. Maydianne is a biologist. She's an award-winning biologist in terms of her research, her activism, her teaching, and she examines the evolution of species diversity across variable environments with a focus on studies of social and sexual traits of black widow spiders. She's a member of many prestigious academies, one of which is the American Academy of Arts and Sciences. She's also a fellow of the Animal Behaviour Society and of the Royal Canadian Geographical Society. And she's a university professor at the University of Toronto, which is the highest academic rank that you can be. So you can see just some of the things that Maydianne has achieved. As well as being a fantastic researcher and scholar, Maydianne does a lot of other really important work in the broader research and in research community in the service of the public. So at the University of Toronto Scarborough campus, where she works, she's the Vice Dean of Faculty Affairs, acting Vice Principal and Dean. And she's also done some incredible work in terms of inclusion or increasing black inclusion in STEM fields in Canada. She's actually the co-founder and inaugural president of the Canadian Black Scientists Network. In addition to her research, and you'll hear a little bit more about this tonight, she's also been part of several documentaries and made a podcast. So she's a very busy person. I don't know when she finds the time to do all of her research and everything else. And we're absolutely thrilled to welcome her to deliver this public lecture tonight. So I'm promised that we're going to take a walk on the wild side. There's going to be deadly spiders. There's going to be sexual cannibalism. And we're going to hear a little bit more about some of her work daring the heights of the Rocky Mountains. So please join me in welcoming and giving a very warm welcome to Professor Maydianne Andrade. Thank you.
Thank you so much. So thank you for that lovely introduction, and I'm so pleased to be here. I appreciate you coming out after hours. I hear there's all sorts of exciting things that you could be doing instead of this, but hopefully you're here because you're here for the spiders. So what you're looking at right now is a female redback spider and two males who are competing to have the opportunity to mate with that female. And I'm going to be talking about, I think I'm going to start with my origin story, because people always say, how is it that a Canadian professor is here studying redbacks? And I have been doing so since the earliest days of my career. And then I'll move from that into the main questions in which I study redbacks as well as black widows. And then I'm going to tell you a bit about science communication. And for that part of the talk, I'm going to come back to this. This is me with members of the Red Trillium film crew and the director, Andy Gregg, about to get in that helicopter and fly into the Kootenay Mountains, with many thanks to the permission to be on the lands of the Dakota in that area. This was to go and chip fossils out of the Burgess Shale. And the Burgess Shale is a famous area in Canada in which fossils of the Cambrian explosion have been captured in exquisite detail. And I've been reading about it since I was a young child. So this here is a book by Stephen Jay Gould called Wonderful Life, and it was full of illustrations of these really weird creatures. I've been reading about it since I was maybe 10 years old. And yet when they came to me to ask me to do this, to come with them to the Rockies, to be host of this documentary, at first I said no. And so part of this story is about how you move from being a scientist to a science communicator. So one of my science communication heroes, at least back in the day, was Stephen Jay Gould. He has now passed away, but he was a prolific writer. He was American, and he wrote a column in Natural History magazine every month, and also several popular science books that I read when I was young. And Stephen Jay Gould actually features as well in my early inspiration in science, because he spoke to a debate about how sexual cannibalism evolved. I'll come back to that after you watch this short video on sexual cannibalism in praying mantids. The gorgeous movie and music is a creative collaboration between Dr. William Brown, who's a biologist, and Phil Hastings. The female is on your left, the male on the right, approaching cautiously. The QR code will take you to the full video if you'd like to watch it at some point. Tension mounts. So that's the female eating the male from just below his head. And eventually there will be not much left of that male. And this is a quote from a book in 1886. Cannibalism has been known for quite a long time. By the end of the night, she had completely rid herself of her spouse, and nothing but his wings remained. Now that kind of observation led to people wondering, how does this work in terms of evolutionary biology? Evolution typically works by increasing the number of offspring, individuals have traits that may increase the number of offspring they have. And if their offspring carry those traits, then that trait becomes more common in the population. How then do you have systems in which a male is killed after one mating, when in most species we see males mating repeatedly? So that's the question, how does that evolve? When I was just an undergraduate, this Professor Ruth Buskirk wrote a paper in which she had an elegant mathematical model. Because if we really understand evolution, we should be able to model it. We should be able to say, this is the currency, babies. This is what they're doing, and here's the alternative ways they could do it. And here's what we predict to see will evolve in this population. And she looked at this idea of males being a mate or a meal, and she found a set of conditions under which males should actually evolve to allow females to kill them. We knew that for females, a male often is a meal in these species. But why is the male allowing it to happen? And people thought that praying mantids were a good system to study this in. But shortly after this model was written, Stephen Jay Gould, again, one of my heroes, said that he doubted it. Do males, for the sake of their genetic continuity, actively elicit or even passively submit to the care and feeding of their fertilized eggs with their own bodies? I find little persuasive evidence for such a phenomenon, and I wonder if it exists at all. So countering the elegant mathematical demonstration that it could be possible, and starting a debate about whether you could have males evolve to allow cannibalism. Part of the debate arose because Stephen Jay Gould did not believe that most of the time evolution happened by this trait increases the number of babies that you have. He thought random factors were much more important. And he said this argument would be an excellent illustration of a curious behavior that nevertheless tells us something about evolution, and it tells us about whether this reproductive success is the critical part of evolution. So it turns out that you can test some of these ideas, this elegant model, but black widow spiders are better for testing it than are praying mantids. And what you saw with the praying mantis, one of the reasons people thought males were complicit is that once the female chews through the male's upper body, it actually releases faster copulatory behavior once the male's head is removed because of the way his nervous system works. But nevertheless, it was clear males were trying to avoid that outcome. They jump away, they sneak up quietly, etc. Black widows, and in particular redbacks, were different. When I started as a master's student wanting to test this idea, Daryl Gwynn, who had done a postdoc in Perth, Western Australia, was heading back to Perth, and a paper had just been published by Dr. Lynn Forster, who is a spider biologist from New Zealand, And she described a weird and wonderful behavior in redback spiders. So Daryl Gwynn said to me, it was about November in Toronto. Our seasons are opposite. We were just heading into winter. And he said, would you like to stay in Toronto and do something different, or would you like to come with me to Australia and study redback spiders? And so obviously I got on the plane. So I got to tell you a little bit about spiders in general and Latrodectus or widow spiders in particular. They are a group that includes many different species that I'm going to touch on. And to understand their strange mating behavior and why it's interesting, you need to know something about their biology. So the female's on the bottom, the male on top. And they copulate. Males have structures on the front of their heads, essentially, those two kind of boxing gloves that allow them to mate with the female. They have two separate organs. Females have an opening on the underside of their abdomen, and they have two separate organs in which they store sperm. So a male will inseminate one side and then the other, one with each one of his palps. Now, in most species in the genus, this is how copulation looks. The male will climb onto the female's abdomen. You can see the diagram on the left and the photograph on the right. And he's inserted a coil at the tip of that palp, and they stay in that posture throughout mating. But in redback spiders, shortly after the male begins to inseminate the female, he flips into a somersault. And he uses that palp as a pivot, and he lands with the back of his abdomen right over the female fangs. And she begins to consume him while they're mating. The male is still able to transfer sperm, and he's also able to pull himself away from the female. And you remember, two copulatory organs, two sperm storage organs to fill. He goes back and he mates with her a second time, and then she kills him during that second mating most of the time. So more than 60% of matings end in cannibalism during sperm transfer with this copulatory somersault. And we also see it in brown widow spiders, another closely related species. And then we found more recently in a South American widow that they don't exactly flip, but the male kind of dangles his legs in front of the female, and she eats his leg while he's copulating with her. So this is essentially behavior that suggests males are single mating specialists. They've evolved to have a behavior that means they only mate once. So the puzzle actually gets even more puzzling. So if we want to try to understand this, we need to see what the balance is for males, because we want to know why males are being complicit, essentially. And if it's in terms of the currency of evolution, then what we expect is males should be balancing other mating opportunities against surviving offspring from the one mating that they might get. And not consciously so. But any male who behaves in a way that results in more offspring will pass that trait on to those offspring, and they'll become more common in the population, right? So this somersault is a trait that evolves. A grad student in a collaboration has recently found that somersaulting is actually genetically controlled by something that is on the sex chromosome. So we know it's something that could evolve in this way. So, more surviving offspring, other mating opportunities. What we did was measure in the field in the lab how many opportunities to mate males typically have. And what we found is that it's very hard for males to find a female. And in fact, about 80% of them die without ever finding a female in nature. They leave their webs, they wander through the environment, and they're exposed to predation during that time. They also don't see very well. So spiders depend on vibrations both to eat and to defend themselves. And males don't have their webs while they're wandering looking for a female. Females are sedentary. So it's a very risky business, and about 80% of them die. So in other words, other mating opportunities are unlikely. If 80% of you die before you get there, the chances that you're going to get to one female and make it to a second one is low. So what about surviving offspring? If you're likely to get one or maybe two matings, but investing more in that one mating will increase your number of offspring more than having a second one, then you could have the evolution of this strategy. And so we also looked at this idea about the number of offspring. And a paper here, actually, Kate Barry was at the time doing research at Macquarie University. And what she showed with her student and another professor is that if you eat a male, you actually have more offspring. So females seem to get some sort of nutrient from the male, and the number of surviving offspring increases. And then together with my collaborators, what I showed is that if you actually copulate longer, it's not so much a mate or a meal, but males are in competition with other males. And they're in competition inside the body of the female if she mates more than once. So if a female mates more than once, she stores sperm from more than one male, and those sperm compete for eggs. The more sperm you release, the more likely you're going to fertilize more of that female's eggs. And in fact, females allowed males to mate for longer while they were cannibalizing them. So this sort of calculus actually works. And it makes sense for redbacks, sort of quantitatively, that this is the right strategy to follow. So I love this because it sort of went back to this debate in the literature. It suggested one of my favorite things in the whole world is that we could predict everything with an equation, a grand unifying theory. This was sort of covered in a couple places that found this idea kind of intriguing, right? Sexual cannibalism in Gould's shadow, it does happen this way. So that is sort of my origin story. That's where I started, and I did study redbacks for many more years. And one of the reasons that I've studied redbacks and related species is because they are very good models for trying to understand some more complicated ideas because of that sexual behavior, at least partly. And so I follow this principle that is, for a large number of problems, there will be some animal of choice on which it can be most conveniently studied. And so the idea here, when people say, why do you do fundamental research on spiders? Why should we care? There are very complicated things that may be important for a variety of other organisms, but that we can't do experimental work on, or we can't really do the kind of work you need to do to understand the dynamics of the outcomes. We can do that on spiders. And then we can see how that scales up to other systems. And one of the things that I'm really interested in is plasticity. And this turns out to be an area where spiders are particularly great, at least black widows. So some of you will know this, genes are plastic, by which we mean you can have the same genes producing different traits in different individuals. The way in which gene expression works can change what adults look like. And so these are water fleas from, actually these are African, and they are genetically identical, they're clones, and yet they look radically different. One of them has spines and one of them doesn't. Spines are expensive to make. Developmental plasticity is what leads to this kind of difference, and that means that their experiences before they become a mature adult, those experiences are what determine whether they grow a spine or not. And this can be adaptive, at least in this species. They've shown it's adaptive because if juveniles smell predators in the water, they will develop spines, which make it hard for the predators to eat them, and if they don't waste the energy. So essentially, as juveniles, when they're developing, these water fleas are telling the future, right? They're telling the future in the sense that they're developing traits that will make them do better in their adult environment. And that's adaptive developmental plasticity. Now, why are we interested in that? Adaptive developmental plasticity might be critical for resilience to climate change. So organisms that are able to do this, not just with spines and not just with predators, may be able to adjust their traits so that they can at least buffer some of these differences with climate change, et cetera. Organisms that can't do that may be less resilient. In addition, we think it may be important for invasive species, helping us predict which species are invasive. Species that are selected to be plastic may be more likely to be invasive because they're able to buffer, again, changes in environment so that when they're introduced to a new place, they can still survive and reproduce. And this latter question is actually what has been the driving force long term of thinking about redback spiders and black widows. Redbacks are invasive. Brown widows, also somersault, are also invasive. Most of the other species are not in that genus. So I'm just going to put this up quickly. If I wanted to go through the evolutionary cascade that's necessary to demonstrate that adaptive developmental plasticity exists in nature, I'd need to measure or do experiments on every part of this cycle. You have to have a system in which there's a changing adult context. So what was good for the parent isn't necessarily going to be good for the offspring. There has to be some sort of information in the environment that a juvenile detects that predicts what it's going to be like when it's an adult and what challenges are in the environment. And then that information has to be used by the juvenile to change its adult traits so that it actually has a reproductive advantage when it becomes an adult. Again, this is all about telling the future. I like to say that because animals are fantastic. But the problem here is that you have to test these things experimentally, but there's also pieces of it that you really can only understand if you go into nature. So the cues that animals actually are exposed to in nature and the types of changing contexts and challenges. And that's one of the reasons why widow spiders are so great. So you ultimately may be interested in asking about what's happening, say, with large mammals. This is a small mammal, that's a cat. But the idea is that large mammals, for example, live through many years. We can't make clear predictions about what we expect in terms of plasticity because they would have to trade off. Their traits have to be good for year 1 and for year 2 and for year 3, and the challenges may be changing. So they're more likely to be kind of generalists across that. But for short-lived animals, that's not the case. So for example, Daphnia live a very short period of time, and in this case, as a juvenile, you may detect something that's useful for you to predict the future and change your development to match it. So this is a good model species because it's short-lived, because you can make clear predictions about adult challenges. Are there predators in the water? You better build some spines. Are there no predators in the water? Don't waste the energy needed to build spines. There's reliable cues of that adult challenge. Basically, the predators eat up other Daphnia, and those chemicals are released into the water. And you can study them in nature and in the lab. So you can do that whole cycle. And I've been working for many, many years, more than I should say, on a variety of different ways of trying to build Latrodectus spiders as a model for understanding plasticity because they have similar beneficial features that make them a good model species. So a little bit more about Latrodectus. Redbacks are one of about 30 species. They're all in the same genus for the biologists in the audience. They look very similar. They have very similar evolutionary history, but they have different behaviors and live in very different habitats. These red dots just show you some of the places they live. And my students and I have studied them across 5 continents over the course of the last 24 years. And so we've worked with a number of different species, but I'm going to talk mostly about redbacks and also western black widows that live on the west coast of North America. And that little cartoon is me before I blew out my knees and got worse vision. Now I just say to the students, yeah, you know what would be really great if you went out every night for a month and watched that web? And I don't have to do it. I used to have to do it. Anyway, so all over the world, just to tell you a bit about how we actually do this work. In the lab, we do rear spiders, and we do have a large number of spiders to do the experiments that we do. We're able to do behavior, dispersal experiments, development and anatomy. We can look at reproductive output and how much sperm is produced by males and how much is transferred. We do running experiments, believe it or not. That's one way of looking at endurance. And then we can manipulate all sorts of things about the environment that would give them cues about their adult challenges. So that top video shows you some rearing. I typically have about 8 to 10 undergraduates and some high school co-op students in the lab doing that work. And then we can do experiments such as the one you see in the lower left, setting up mating experiments under macrovideography because the spiders aren't that big. And that lets us do this work at scale. We also are very interested in the signals or the cues or the information available to spiders. And so we have done work on vibrational signals, very important through the web. They also use chemical signals a lot, both through touching the individual's exoskeleton or the web, but there's also airborne components. And we're particularly interested in those airborne components that can be detected by males at a distance. And so this shows you some of the work we do. So that little T bar has been dipped, the filter paper has been dipped in pheromones of different females of different stages, and we look at which way the male goes and whether he lays down silk like this one's doing as an indicator of interest or ability to detect that female. And then the other image on the right is actually vibrometry, it's called. You're hearing the dancing of a male spider. So male spiders will court on silk even if a female's not present. So that little chamber is a respirometry chamber, which means we can measure how much CO2 and O2 are inside the chamber in very minute quantities. And that lets us see the energetic cost of a male engaging in this signaling. What you're seeing on the bottom is a laser vibrometer, which is like a little laser beam we shine on the web. So at the same time, we can measure the amplitude of the signals the male is producing, which also lets us measure energetic output. So all these things are important for understanding things about sexual selection in the field. Back to nature now. We also do field work. So these are a series of cages set up across a beach, which is one of our field stations areas. And in these natural populations, we use what we call pheromone traps. So these are full of silk produced by a female, and they are releasing pheromones that actually can move as far as 50 meters down the beach and be detected by males who then travel through the environment, and this is the mate searching I was talking about, trying to find females. So this little guy will be wandering across the sand, walking onto vegetation. They do something we call scenting. They detect pheromones using sensory hairs on their legs, and they will go to high points and actually kind of wave them around, and then find a pheromone plume, essentially, and follow that down the beach. So we're able then to see how quickly males get to webs, what kind of females attract more males, et cetera. And then finally, for field work, we are able to track individuals over the whole season. So we have marks on individual webs, and myself and my students can go back to those webs every night and see who's there. And we identify individuals, 8 legs, 5 colors of paint makes a very large number, so we can identify them as individuals and see who goes where in nature. So one of the reasons we're interested in adaptive plasticity and thinking these could be a big model, a good model, is that there's a huge amount of variation in nature within the same population in male size. And so what I'm showing you here are male western widows, which differ in size by 5 times from the smallest to the largest. And that distribution is male redback spiders, over 200 that were collected in Perth, and the smallest is 1.5 milligrams, and the largest is 10.1 milligrams, a difference of 7 times. Now, because people look at spiders and say, oh, who cares? I want to show you what an analogy here, right? I could not find footy players from Sydney, sorry, with this difference. But you would look at this difference in the height of men and say, that is incredible, an incredible difference, right? And that's only 1.2 times. So finding organisms that differ in size by 5 to 7 times is actually quite a lot. Needs to be explained, and we thought plasticity might explain it. So together with now Associate Professor Michael Kasumovic, my first PhD student, he's at the University of New South Wales, we started playing with this idea of plasticity and thinking, okay, what affects the challenges a male has to find a female, that mate searching that has very high mortality? And we recognize that it would be affected by the density of females in the habitat. Sometimes females are very close by in a high density population, and males don't have to move far to get them. Sometimes they're in a low density population and females are far flung, and it would require a lot of endurance to get to those webs. In addition to which, those few females would be under a lot of competition. There'd potentially be other males that are competing for access. So we thought at a high density, males might shift into a scramble competition phenotype or trait set, like the horns with predators. And at low density, male phenotypes might shift into an endurance type set of traits. And the way that works for invertebrates, like spiders, is for a given amount of food, if males develop quickly, they will be small. And if they take more time so that they can get larger and have more resources for that traveling, they need to take more time developing. So we expected to see this difference triggered by density. Rapid development in small size, get out to those females quickly, or large size and slower development. Now, why do we think density was important? Well, we knew that there was adult variation in density in nature. Females produce up to 2 egg sacs every month. This includes redbacks, about 300 spiderlings per egg sac, and they can do that for a year after mating once. And they disperse by ballooning. So you've got a whole bunch of spiders flying through the air, and this is a lot of black widows coming out of an egg sac, meant to be scary. But they also disperse by ballooning, which means all of these spiderlings land in unpredictable places. So this is from a science experiment a few years ago, and go. So you release a little bit of silk, and then it's caught by the wind. And that means, again, the challenges your parent experienced may not be the challenge that you experience because you have dispersed unpredictably to new habitats. You can see this in the field with thanks to the Sayout First Nation who allowed us to do this work on their lands. Dr. Catherine Scott, Dr. Sean McCann stayed out there for an entire season. And in this study site, black widows, western black widows put down their silk under logs. And what they did was do what I showed you. They marked the webs, they marked males and females, and they found that if you look over the course of the season, which this is our summer, July, August, September, the number of females, receptive females on the beach at any one night is always sort of around 15 or under until the very end of the season. The number of males peaks at some point and then drops off again. And I just want to point out these scales are different. So for the male, it goes up to 250 males on the beach. For the females, it only goes up to a maximum of 30. So in other words, males are always present in higher numbers than females. But at some times of year, there's barely any females, and other times of the year, there's a lot of them, and they're high density. Now, why do males have to scramble to be the first one to reach a female? Why is it worth doing that when you're in a high-density population? Because the first male will fertilize almost all of the female's eggs. So I showed you the copulatory organs, which you can see here again on the left. And there's a small part at the tip of this coil that's inserted in the female that actually breaks off and stays inside the sperm storage organs after mating. So this little hair-like structure here is a plug. A female can mate again, but if that plug is in place, the next male won't fertilize any of her eggs. Can't get access to this sperm storage organ. And we've shown in redbacks, in western widows, and in brown widows that males do this. So if you're the first male to get to a female, and if you copulate twice, and if you place 2 plugs, game over, right? So you scramble to reach that female. But it's not just a race for adult females, because in fact in redback spiders, and we've now shown in brown widows, and actually black widows as well, males can mate with females at the last juvenile stage just before they molt, just before they shed their skin and become an adult. In these spiders, the top picture shows you a juvenile, a late instar, we'll call her subadult female. And towards the last few days of this instar, there is a cover, the exoskeleton, over the genital openings. But underneath this, the sperm storage organs are completely formed. And they'll be retained through the final molt. So males have actually evolved to recognize subadult females, tear open a very precise spot just above the opening to the genitalia, inseminate those females, and then they molt and begin producing fertilized eggs immediately after molting. This is more likely to happen at high density because males, subadults don't let off any cues, but adults do. And as subadults and adults are close by at high densities, males end up encountering subadults. Now, before you shudder, we've done experiments that show that this is beneficial for females. Female black widows are sometimes in a bad spot and suffer long delays to mating because some males never find them. So for a female to not pay the cost of mating and waiting for a mate as an adult is actually beneficial. Females produce more offspring if they start producing them immediately after becoming sexually mature. This is in nature, remember, so you could be killed at any point. So this, we think, is actually beneficial for females. And it does occur in 3 different species. So the race, the scramble, is real. When females are nearby, males scramble. But if females are far away, larger males do better. So at a low density population, what you're seeing here is the male on the right is a large male. The male on the left was actually mating. He's a small male. And oh no! Sorry, I shouldn't laugh. But this is very rare to see. Fatal fighting is actually rare to see in the animal kingdom. It's usually kind of ritualized. But we do see fatal fighting when 2 males are competing in this way with black widows, particularly if there's a size difference. And looking at a large number of studies, overall, large males win about 75% of these competitions. Small males win about 25%. So there is a difference in success as a function of male size. If you have to move a long distance, you need to be bigger and have put on more resources to allow you to make that trip. I didn't mention that males don't eat when they're adults. So all the nutrients that they have available to them to make this trip depend on how much they ate as a juvenile. So again, larger males win direct competition, which takes more time in development. So we did a series of experiments, and I should say two things. First of all, I use the royal we when I say we. It means I said, that would be really cool if you would do that, and the students typically went away and did it. And so I'm putting their pictures up so you know who actually did the work at the bench. Also, for those of you who might be biologists and want to actually see the data, I'm not going to show much data, and I put up the citations here as well, and I believe it's being recorded, so if you wanted to take a look at some of these, you can. Okay, so what we did in Western widows and the Australian redback was to ask, if we manipulate the cues that we think males use to detect the density of a population of females, will they change their development and end up as we thought they will as adults, either in a scramble competition morph or in a direct competition and endurance morph? And for various reasons, for Western widows, very seasonal, as I showed you in that graph, we predicted that it would be cues of the time of season that males would be responding to, and so we manipulated day length. And then for redback spiders, seasonality is not quite as important, and we thought it was more important that there was spatial variation that they could detect through pheromones. So we did experiments exposing subadult males, males in their last instar, either to differences in photoperiod or differences in the amount of pheromones that they could smell as they developed. And what we found was actually what we predicted. And again, that's all those papers that I'm not going to show you the data for. You just have to trust me. But we did find this. So there was a significant difference in development time. If males can smell females while they develop, no physical contact, no visual contact, no vibrational signals, just pheromones from a distance, they accelerate their development, and the result is they're smaller when they become adults. The same is true if you give them seasonal cues that indicate the time of year when females are present at high density towards the end of the season. So day length changes over the course of the summer, especially in Canada, believe me. It's dark all the time right now. Not all the time, but most of the time. Okay, and then the other thing is that when the cues indicated a low density of females, so either no pheromones or seasonal cues indicating that it was early in the season, males took longer to develop and were bigger as a result. So this was a very exciting result. This sort of plasticity would allow us to look at a large number of different traits, but we hadn't finished because we also had to know whether or not males actually did better when they shifted their development in this way. And so what we did was laboratory studies to ask this, as well as field studies. And in the lab studies, so this thing on the left is actually a racetrack. If you put a male in this racetrack and touch him gently on the back leg with a paintbrush, it simulates a predator trying to attack, and the male starts running and running and running. And it turns out redback spiders can run for like 5 hours. It's really remarkable and frustrating when you're trying to figure out when they'll stop running. And so if you do this experiment with large and small males who were reared under the conditions that triggered that developmental shift, what you find is that the small, faster developing males run faster and for longer. They're less likely to stop. The bigger males stop more frequently and just don't run as long. So it looks like a scramble morph. Male becomes mature and is able to run to females, literally run to females. And then we did field enclosure studies. And so we're focusing mostly on whether small males have an advantage, because in most systems we know that large males can do quite well. The question was, are there conditions under high density when small males would do well? And so what we did in a field study and also a field enclosure study is in the field, we put out silk inside a pheromone trap, and that was simulating the maturity of a new female who is unmated, because those silk traps were releasing pheromones. And then we tracked which males were attracted, and we measured them, and we did various things to measure their traits. We also did a field enclosure study here in Sydney, where we put out a grid of females at a density that was a high density relative to what we found in the field. And then we released males, large or small, but we released them with the small male being released just a little bit earlier, actually just 1 day earlier. And what we found is if you release the male 1 day earlier, which is very similar to the developmental shift, the male gets to the female, mates with her, and then the female stops producing pheromone and the large males never find her. If a female is not producing pheromone, she's invisible to the male. And similarly, in the field studies, what we found is that if we put out our pheromone traps in high density fields, some populations had very high density, the small males came first. When we put those pheromone traps out in low density fields, large males came first. So we think that we have here a very interesting, oh yeah, those are my small and large males, system where we can manipulate these things, where spiders can be a model that allow us to probe when do you see the evolution of plasticity, when do you not see the evolution of plasticity, how does that affect invasiveness? That's one of the things we're really interested in now. And again, just to remind you, it's also powerful because we have 28 species that are not invasive and 2 that are. And the 2 that are 2 that we know definitely have this kind of plasticity. So there's lots of questions left to ask, but we feel like we're moving towards a place where we can do a comparative study that would allow us to pull out some of these details. So how do we go from that to this? Very kind of roughly and clunkily, but bear with me. So as I said, I was doing this work for 24 years. It involves a lot of public talks like this. It involves working with school children. It involves going to zoos with my spiders. Everything from kindergarten to when my kids said, stop coming to my class at about grade 9. And so I'd spent a lot of time, as well as teaching. So I used to teach evolution, animal behavior classes of 500. I spent a lot of time translating science for different kinds of audiences. And yet when they came to me and said, will you be the host of the nature of things in this, go to this place that I've always wanted to go to, I said no. And it's even more remarkable because the particular show that they asked me to be on, The Nature of Things, is actually the longest running TV science documentary series in the world. It started in 1960. And David Suzuki was another one of my idols. He hosted The Nature of Things for most of that time, just retired 2 years ago. And in Canada, he's a very famous activist and climate change activist. So I idolized him. I really wanted to go to the Burgess Shale, but I said no. And I realized in reading about David Suzuki that one of the challenges was, as a scientist, we're taught to pack as much data as possible into every talk we give. Graphs and statistics. We're taught to be very precise in our use of language. And that is not what you do with science communication. We want to talk to people and have a story and make sure that that story is interesting and that people can understand it. Just like if I were to go to my brother who's a mechanic and say, hey, how does this car work? I don't want him to tell me about the size of the bolts and stuff because I don't understand what he's talking about, right? And so this kind of storytelling is risky for scientists, or it feels that way, or it used to feel that way. And David Suzuki talks about being scorned by the scientific community when he started doing this. So anyway, I decided, you know what, I should really do it. So I ended up here, got in the helicopter, went up to the Kootenays. This is our campsite, and there was a large group there from the Royal Ontario Museum, and they were been studying this, this fossil site for many years. Again, this is a site in which the very earliest forms of animal life have been preserved. And that arrow shows you where the dig site was relative to our camp. So it was up around the corner there. This is already on the side of a mountain. That is a bear fence. It's electrified. Our tents were outside the bear fence because we weren't part of that group, but that's okay. I didn't know that till I got there. But one of the things about this trip is that you had to climb up every day to that high site. And oh yes, did I tell you I'm afraid of heights? I'm definitely afraid of heights. So I'll show you a little bit from that documentary.
It turns out they didn't really make them for people with hair like mine, but I can basically get it on. And then I'm protected like everyone else up there. Very important. So let's see how we do for the steepest part of the hike.
Here we go.
All of this slippery shale has come off rock faces above. Some fossils tumble down the ski slope, and those are the first clues the museum team looks for. They find the right shale face and start chipping off samples. Once they see critters, they establish their quarry. Hey guys.
So what you saw there was what happened for the first 3 days. I felt very comfortable. We were actually in the middle of a forest fire year. Then it rained. And what you just saw is where we were. I was terrified. But what really allowed me to do this is that the first 3 days we were socked in with that smoke. And I couldn't see how steep the mountain was. And I kind of feel like it's the same with science communication. You just get in a little bit at a time, you find your feet, and then you start seeing how high you are up on the mountain. And that's showing you going around to the other dig site that we went to. So in the end, it was a very rewarding experience. Not only did I get to narrate this amazing documentary, but I was taught how to chip out fossils from the shale, and they found a new species while I was there, which was pretty thrilling. And then when it was over...
Sex in bugs is fascinating, gruesome,
When it was over, they said, "What's next?" And I said, "Oh, bug sex, obviously." And the director said, "Okay."
And most people know nothing about it.
They do things like we do, but they do it in ways that are really different than the way we do it.
There's just a wealth of different diversities out there that lead to the evolution of all these weird and wonderful ways of doing it.
It's just a little blob of sperm, and he threads it into her reproductive tract.
It actually is heart-shaped.
That's what he was giving her in exchange.
Okay, so you get the idea. I'm sorry I put the QR code up there, but I don't know if it's viewable in Australia or not. But I didn't actually know he was going to call it bug sex. I thought we were going to change the name, but he didn't. And the point is that it turns out people are interested in this type of thing. And you can talk about general evolutionary theory in ways that are engaging and interesting and comes along with some really lurid videos. So with that, I think I'll end, and I'll be happy to take some questions. And I want to make sure that I thank again the Stay Out First Nations, whose lands we worked on Island View Beach. And in the Kootenays, we worked on the Tunakha Nation's lands, as well as Takamanawak. And I still can't quite get that H. And so that was very generous of them to let us use their lands. They're very interested in learning about the organisms there. I want to thank the amazing members of my lab over the last many, many years, including Isaiah Box, who's actually going to be coming here next week to collect redbacks. If you have any on your property, I have a notebook up here, and if you want to give us your contact number, we'll come and take them away for you. And then the approximately 200 undergrads and counting that I've had over the years, and of course the redback spiders. So thanks very much for your attention.
Thank you so much, Maydianne. I think it's fantastic that you shared that you faced your fear of heights when some people might have faced their fear of spiders to come here tonight. So it goes to show that we all have things that are more or less scary for all of us. Thank you for such a wonderful talk. As Maydianne mentioned, she's very happy to take questions from the audience, and we have some microphones here. Does anybody have a burning question? And could you just wait for the mic to come to you so that everybody can hear you?
So when you had the small males and you said that they can run for up to 5 hours, whereas the big ones would stop frequently, did that affect their final molt, making them run like that because of the energy reserves used?
That's a great question, and I didn't explain it very well. That was as an adult. So we exposed them to the pheromones, we let them molt, and then all the tests we did was to check out what their adult traits were like. Thanks.
We'll take one question here and then one up here, Scott, if that's okay.
Thank you very much. That was fascinating. I'm not a biologist, so I probably going to ask a very silly question, but what's the reason why there are so many more males than females? Do they just a lot more born, or do the women die off? Yeah.
No, that's a great question. Every single talk I give, people ask me that question because it's a pressing question when you see that sex ratio difference. In each egg sac, we actually think it's 50-50 male and female, but females might take the whole season to become sexually mature, whereas males mature in only about a month. Because they are smaller, they don't take as long, they don't go through as many molts. And so what you actually have is some females overwinter, so that, at least in Canada, overwinter, here too, I think. And then they start producing egg sacs right at the beginning of the season, and the egg sacs start developing. So you end up with multiple cohorts of spiders growing, but the males keep maturing quickly, and the females take much longer. So that's why the sex ratio is off kilter in nature. And then on top of that, you know that males must be even more numerous than that because a lot of them are dying before they ever get to a female, and those we counted sort of on females' webs mostly. Thanks for that.
Thank you for your talk. I really enjoyed it. I love the idea that you're doing all this research, but I just want to know, what are the practical applications? How are you going to use this research?
Yeah, so I mean, that's a really good question. I think, so there's two questions. One is there's no direct practical application. It's kind of like ballet. That's one. We learn about the world, it enriches us. But I really do think we need to understand plasticity. It is a pressing question whether this will help us to predict. If we can predict which animals are plastic, do we know which ones to have higher levels of vigilance against in terms of invasiveness, right? So redbacks, for example, are repeatedly introduced to Japan. They're in Japan, they're established, and they show up repeatedly on shipments, so they keep being reintroduced. They do perfectly well there, despite having really different climate. Western black widows are repeatedly introduced all over North America in grapes. So people are always saying, I found a black widow in my grapes. I had it in the fridge for a week. How come? And females can survive a week in the cold. So nevertheless, we don't see introductions. We don't see Latrodectus hesperus, the western widow, establishing all through North America. So the question is, what's the difference between those two? And can we leverage that information to help us understand what's happening with other organisms? As you may know, invasive insects are actually a significant challenge, especially in places where orchards, et cetera, are affected by them. So hopefully that helps. Oh, and then there's the other side. Sorry, wait. Some people milk black widows and use their venom for research because it causes the release of chemicals at the nerve endings that can be used to manipulate different kinds of things. And they've learned how to create silk in bulk. And they were actually running shoes made out of spider silk. It's black widow silk is extremely strong, stronger than Kevlar, which is in bulletproof vests. So it has these great properties. It doesn't stick to wounds, so there's applications for using it on wounds. Not my research, but maybe the 100,000 spiders can be useful.
There's actually some research here in Australia between Professor Glenn King, who's at the University of Queensland, and some of our colleagues in the School of Chemistry. And others around Australia that's looking at how different venoms could have applications for treatment of stroke and other things like that. So there's a lot of secrets being discovered from these substances
great.
Too. Do we have any more questions from Maydianne? Just while Scott and Cassie, you could help us find the mic, I'm going to sneak in a little question if that's okay. I was wondering, in terms of short-lived species, as you describe them, having perhaps a greater degree of plasticity, and that they're, the sort of, because their life cycle is so short, evolutionary time is maybe sped up. Does that mean that smaller or short-lived species are going to be more likely to survive or be resilient to climate change than larger or long-lived species?
Yeah, it can be. So I should have said, so that sort of cycle, the short-lived and information being important from juvenile stage to adult, that is an important part, and these short-lived, rapid developing species have that. But they don't always have the other things, which is there's not always information in the environment that will cue them to what's going to happen to them as adults. And it's not always the case that there can be a shift like the one I showed you between two different kinds of options to be what type of an adult you could be. But having said that, the second part of your question, yes, short-lived, rapidly reproducing, multiple generation organisms evolve faster potentially than organisms that take much longer. But unfortunately, climate change is happening at such a fast clip that there's not time for most organisms, other than, say, like flies and cockroaches, to sort of catch up with it. But we do think that plasticity could allow them to survive a wider range of conditions, which might give some organisms time to catch up evolutionarily, right? So actually, if you start out being able to survive in this condition, and some organisms have a genetic trait that make them slightly better at that survival under plasticity, then maybe those traits can evolve over time. But we're in a race against climate change, and I don't think we're winning. Yeah.
So one question over here.
Thanks, super interesting talk. I have a question about the functionality of being bigger when there's lesser females around, and whether that's also, you said it's a mobility thing, they can move further, but being larger and having more surface area for chemoreception on limbs, which is just amazing, does that allow them any advantages in locating females in an environment where these pheromones are lesser distributed?
That's a great question. So they would have more sensory surface, you're right. And they may also be able to then detect females at a longer distance. We haven't done any experiments to look at that yet, though. What we have done is demonstrated that in addition to the, so this is kind of, there's exploitation of those good at finding female males. In the beach in California, when a male finds a female, all spiders, while they walk, they leave a drag line, which is a little line of silk behind them. And so males that are good at finding females, including larger males, will leave this trail of silk as they head to a female, and other males use it like a highway. They actually get on the silk and follow it as fast as they can to get to that female. It's very unusual to find males seeking out competition, basically. But it's because, again, they're so hard to find. So it means that if larger males were able to do that, they could also be exploited. That ability could be exploited by smaller males.
Hi, I'm just here as a member of the public. I heard you on ABC this morning.
Oh, so you heard some of this then?
Yeah, just a little bit. I was just very fascinated, and that was amazing. Thank you. So I'm not a scientist, just purely out of interest. Just a question. So, have you observed? I imagine in the lab it's controlled, but if you're observing the spiders in the wild, have you seen any cases where the woman will eat the male outside of a sexual? Or, I mean, like in that image, the first video you showed of the one male who won and was mating with her and a second close by, will she knock him off as well just for fun, or like extra resource and protein, that kind of thing?
Yeah, that's a great question. And I love it when people say I'm just from the public, because really the observations, the same ones emerge no matter what audience I have. And you're absolutely right. So the thing that I didn't talk about at all is the female side of this. And lately, not lately, but in the last few years, we've really been focusing on that. It was harder because females are more long-lived, but there's a few things. Females will knock males off wags, and they can do that. So when you saw that first video, the eye's drawn to the male. But like I described to someone, the female just sitting there, it's like I was sitting in the front row here just bristling with knives and very angry, and you came up to me and I just sat there. Right? I have other options. And so the female does too, and yet just sitting there is actually a decision on the part of the female. So you're right, in some cases, she will knock males off webs. In some cases, she will cannibalize males after they only copulate once, and then she leaves the other organ open. And they're more likely to do that when, they detect more males in the environment. So when females are developing, if they smell male pheromones, they will be more likely as adults to kill a male after one copulation because they know there are lots of males around to choose from. And that way, instead of being plugged on two sides, the females could choose a second male and then at least have mixed paternity of her offspring. So thanks for that question. Whole nother line of research I just brought in like 5 minutes.
Any more questions from the audience? I thought I saw another hand. There's a second question down here. Is there anyone who hasn't had a chance to ask a question? There's a person. Scott, do you mind going to the person with grey t-shirt first, and then we'll come back down to you? And if there's anybody else, just give us a little wave so we can find you.
Hi, thanks for the amazing talk. That was really good. My question is a bit kind of general, but like when you're working with the spiders in the lab and in the field, do you have to worry about getting bitten and venom and stuff? Is that like a problem or?
Yeah, it's a problem. You might have seen my assistant was wearing gloves. So we wear gloves because theoretically it will slow down how long it will take the female to bite you. We have a whole set of biosafety protocols. I have insurance through the university for anyone who signs off on working in the lab. They have to watch biosafety with Black Widow videos. We have lab coats that close that cinch at the wrist, like all sorts of things. Nevertheless, I have had one student get bitten. It was someone who was very practiced with working with Black Widows, but was ignoring the safety protocols. It's like people say, once you get comfortable with something, that's when you're going to get hurt. Black Widows are not aggressive. They will typically try to get away from humans unless you're taking their egg sacs. Females are extremely maternal. They hang onto their egg sacs, they sun them in nature, and they will bite metal forceps when you're trying to take the egg sacs out. So anyway, the student who's very comfortable with Black Widows was taking egg sacs out for our maintenance of our population with no gloves on. The female got out, climbed on her hand, and she let it, and it bit her. And so anyway, I was at home that day. I got a phone call. Maydianne. I was like, yeah, what's up? So-and-so's been bitten. Okay, did you call 911? No. Is she sitting down? No. Did you put ice on the bite? No. Okay, remember that red binder by the door? Yeah, so anyway, it ended up being fine. She said it was remarkably painful. It was like having a really, really bad flu for about a week after one little bite. But she was seen at the hospital and sent home to just sort of manage the symptoms. So typically, only people who have a very negative effect will get the antivenin. There is an antivenin. No one's died from redback bite for quite a long time, but it can be very unpleasant. Yeah, so thanks. Long answer to a short question.
Just a question at the front here. Maydianne, is it just the females that are venomous, or are both males?
Males are venomous too, but their fangs are quite little. And so they're not likely to be able to pierce our epidermis very easily. Okay. That's our theory.
So just as the other question, you said that you collect spiders from urban areas. Have you found that spiders, if they're exposed to, say, pest controls or such, like pest controls and poisons, do they develop differently? Are they more likely to be small or large? And just off the top of that, are urban spiders different when you collect them to those in nature?
That's part of what Isaiah is here to study, is whether there's differences between populations in urban environments versus natural ones, and whether the urban environment allows them to tolerate a wider range of conditions as well, because they're shielded, sort of, both by heat island effects, so the cement's soaking up heat, and then overnight it doesn't get as cold in places where it's colder. And also by being shielded from moisture, which can actually be damaging for them if there's too much moisture. So that's one thing. I haven't actually seen any effects of pesticide. I haven't looked at that. I know some people have worked on that in other spiders, and it does do things like affect how they build their web. It can affect how many offspring they have. It can affect their behavior, as in they're a bit slower and kind of clunky, so it's not great for them. This morning on ABC, I was talking about the fact that actually their egg sacs are largely impervious to pesticides. So we've tried, other people have looked at that and Black Widows and found that you can't really, if you spray a web, you'll kill the female. Her egg sacs are perfectly fine. And depending on the staying power, then 200 babies come out, or 300 a couple weeks later. So it's a hard thing to kill a population of redbacks. I don't advise spraying them, really. You're better off calling us and we'll come pick them up.
Yeah, of course.
So, yeah. Thanks.
Do you mind if I just, following off that?
Yeah.
I actually used to work in pest control. I like bugs, that's why I don't work there anymore. But we had a lot of redback calls from service stations. They'd particularly be on petrol bowsers. Would that affect the way the pheromones are transferring and found by males if there's strong fumes or something like that? Is that possible to observe in nature as well?
That's super interesting. I don't know. I mean, so I collaborate with identifying the pheromone, and there's a chemical that's on the web, and as it breaks down and volatile is released, I'm really curious about whether it can catch a ride on something or whether it would actually suppress it. I don't actually know that. It's cool that they hung out on the petrol pumps.
Yeah, frequently. It was where we got most of our redback calls from.
Oh, thank you. Great tip. Okay.
I think on that note, when we might have launched a new research project.
Collaboration, yeah.
We might wrap up this evening's discussion. It's been wonderful to host you this evening at the University of Sydney. This has been a co-hosted seminar between the School of Life and Environmental Sciences, or SOLS, and the School of Chemistry, together with the Faculty of Science. So thank you all for coming, and please join with me in thanking Professor Andrade for a wonderful lecture, and to wish her well with the rest of her studies while she's in Australia. Thank you.
Thank you so much. Thank you. Appreciate that. Thank you.
Thank you.